All right, these are your notes on protein synthesis, and we're going to use this document as you go through the notes. I'm going to be flipping through a couple of different presentations to get you the documents. If you would like to get full credit for this, then it's really important that you answer these questions on the side as well as fill in the notes. But looking at just our goals, we want to understand how the structure of DNA is different from, or the structure of RNA is different from DNA, and we want to understand how the information in DNA is used to make a protein. So if we start here, um, protein synthesis is really all about decoding our genome, making proteins, decoding the genome to actually make proteins. Synthesis means to make things. So what do we know? Metabolism is controlled by enzymes. Enzymes are proteins. DNA contains that genetic information to build proteins. DNA is found in the nucleus. Ribosomes are not. This is where we make the proteins. And how can we then get DNA to be used to build our proteins? So if we look at again, the larger picture here, we have DNA that is found in the nucleus, and that's the plan for making the proteins. But the ribosomes are made here outside the nucleus, or the proteins are made in the ribosomes outside the nucleus. So the question is, how do we get that DNA right here in the nucleus to the ribosomes? And the answer is the RNA transfers it. Okay. So when we look at, again, sort of this central dogma of the flow of genetic information, DNA is replicated and then transcribed, right? DNA replicates to make more DNA, and DNA is also transcribed to make RNA. RNA gets translated to make our protein, and the protein causes the trait. So this is sort of the central dogma of genetic information in a cell. So looking at just some language barriers, DNA and proteins are not written in the same language, and that's why we have the DNA and RNA, right? The monomers of a DNA are, and RNA both are nucleotides, but the code is a little different. So we need a translator to make the information from DNA, to make it make sense for the transfer RNA at the ribosomes. And so, we use messenger RNA to kind of do that for us. So what does RNA look like? Traits like eye color are determined by proteins that are coded by DNA. And again, the RNA is the translator here, right? It's the decoder ring that makes the information in the DNA understandable. And we need to get those instructions on how to make the proteins from the nucleus to the ribosomes you saw in the animation. So the RNA is going to help us do that. And just to back up, there are three types of RNA. All right, we have messenger RNA, and this is the information that is going to run the information from DNA in the nucleus to the ribosomes. Then we have ribosomal DNA, or ribosomal RNA, rRNA, that is part of the ribosome that reads the messenger RNA message. And then we have the transfer RNA, that is the translator that puts the amino acids in the right order to assemble a protein. So we have to remember that the DNA to make proteins is, or the code to make proteins is in the DNA. All right, proteins are made up of just a bunch of amino acids, and we get that from the food that we eat. So when we eat food that contains protein in it, our body has to rearrange those amino acids in the right order to make human protein, and we know how to do that by the code in the DNA. So if we kind of want to look at what we're sort of dealing with here, we have ribosomal RNA, okay? Then we have messenger RNA that looks like this, okay? It is single-stranded. It has the same nitrogen bases with the exception of what we call here uracil. There is no thymine, there is a uracil, and messenger RNA is divided up into 3 base codes called codons. Right, that's a code for a specific amino acid. So this code is lined up or made from the DNA, and then it's going to go to the ribosome and put together our protein. Again, each code codes for an amino acid. So the transfer RNA, it's your RNA strand, but it is kind of folded up on top of itself, and it is the amino acid carrier. So your transfer RNA goes and picks up the amino acid from the food that you eat, takes it to the ribosomes, and put these amino acids in the right order based on the code. And that's the bottom 3 bases is called the anticodon because it matches up with a codon. So the codon is the messenger RNA, and that tells us which of these amino acids should go together. And we know that by this anticodon. No, that'll make a little more sense when we get to the actual steps on how to build our protein here. So looking at just RNA structure again, we have RNA and DNA contain the 4 nitrogen bases, but instead we have uracil instead of thymine. We'll do some practice with in class about how this all works. But uracil forms a base pair with adenine, just as thymine does it in DNA. So in RNA, U pairs with A, but in DNA, T pairs with A. So that's just a little different. And we'll get the hang of how this all sort of works together here after the notes. But looking at the steps. So the first step is transcription. The second step is translation. And the third step is protein folding. So transcription here. We want to rewrite the DNA and the messenger RNA so that the instructions can leave the nucleus. All right, that is sort of step number 1. Okay, transcription. All right, the process of making RNA, messenger RNA out of DNA. Again, within the cell, the enzymes make an RNA copy. All right, we're looking at enzymes like helicase and polymerase that are going to unzip the DNA and then put the nucleotides in the right order. And this nucleotide sequence, rewritten as messenger RNA, acts as, again, the messenger. It is taking the message from the DNA to the ribosomes to put together our protein, but it's not complete. Right, it's not complete information for building the protein. Right? For example, if we were going to here's the DNA. If we want to transcribe that code using the RNA base pairing rules, DNA can be easily coded. So we have the code here, but look what we have here. There is a T in DNA, which matches with A in RNA, but no T in RNA. A in DNA pairs with U in RNA. So now we have our 3 base codes, every 3 bases codes for particular amino acids, and then we move on to the translation part. So again, just to show you a little animation of how this works, I'm going to kind of flip through some slides here. All right, transcription. Here we go. We have DNA that unzips, and we make a code again. We have the DNA on the inside, messenger RNA on the outside. And now we can take that messenger RNA copy to the ribosomes, and we are all set. So again, we've now made our copy in the nucleus, and now we can go right to the ribosomes to where we can start lining up those amino acids. All right, that's transcription. Translation is a little different. Okay, translation here, we are taking the RNA, the messenger RNA, and translating that code to put the amino acids together to make our protein. We are converting that messenger RNA into the language of proteins in the ribosome. The ribosome is like it's just a helper. It's going to kind of move along that line of messenger RNA. So just to give you information about the genetic code, okay, we have 64 codons, right? Every three bases on the messenger RNA makes up a codon. It's a set of 3 bases. We have 64 of them, all right, which represents about 20 total amino acids in our body. So all organisms use the same genetic code, okay? DNA is DNA anywhere we look. You guys looked at banana DNA. We could do strawberry. It all looks the same. It's just different letter combinations give us the different proteins. But all organisms use the same genetic information. So UAC codes for the amino acid tyrosine in bacteria and birch trees and bison in all living things. So that stays the same. So we when we look at a map, this is a map of all 20 amino acids, we start with a start codon, AUG, and that tells us, let's start building our amino acids. Then we also have a stop codon saying we're going to stop building. This is all for life, and we'll get used to using this code as we practice throughout, but just know there is a way of kind of mapping this all out as we go along. And again, this is for all of life. The strongest support for a common origin from all life. We all came from the same materials, and the code is redundant. I mean, several codons make up each amino acid. So here, there are 3 different ways to get isoleucine, the amino acid. There are 2 ways to get this amino acid. There are 2 to get lysine, okay, aspartic acid. So we have lots of different ways to get different, here's leucine, to get different amino acids. All right, so here's the process. We have again, we have the DNA, we got to copy it. So we make the messenger RNA, and we got a 3 base code that makes up the codons. Codons are read by the ribosome 3 bases at a time. Okay, so again, more on the codons. The codons are just not a code for amino acids. They provide instructions for assembling the proteins, like the stop codon tells us when to stop on that line. The start codon tells us when to start. This codon codes, for specific amino acids. So let's take a look at how this all kind of builds together, and then we'll kind of go back. Translation. All right, so the first step is we have the messenger RNA at the ribosomes. Remember, we went from the nucleus. We first copied our DNA in the nucleus to make messenger RNA. It is at the ribosomes. So at the ribosomes, we got to, translate that code. So the messenger is lined up. We got the 3 base codes. This is transfer RNA that matches up with the messenger RNA, and it's kind of tugging along. It's tugging along those amino acids. So as it's doing it, the ribosomes are pushing it along. And with each scenario, you've got a bond that forms between the amino acids, and then this transfer RNA kind of gets kicked out. It detaches from the amino acids, and then it's going to go find more amino acids to bring back. Then this one's going to detach from the amino acids, and it's going to bring more amino acids back. So once the bond is formed, the transfer RNA detaches from the amino acid to bring more back to form this chain. And that's called a polypeptide chain of amino acids, and that folds into a useful protein. So looking at, again, going back to our notes here, the transfer RNA is the key to the translation. It transports all the amino acids to the ribosome. Each transfer RNA molecule attaches to only one type of amino acid sequence because of its structure, right? That's done by the base pairing called the anticodon, right? So you have at the ribosomes, which is made up of our RNA and proteins, the messenger RNA lines up right here. Here we go. Here's the messenger RNA, right? Here's the transfer RNA, and then here's the site up here where it's going to tug that amino acid to build our polypeptide chain of amino acids that will eventually fold into a protein. So again, first step, messenger RNA reads the code. Here's the messenger RNA. Here's the transfer RNA. The transfer RNA brings the correct amino acids. The amino acids assemble a polypeptide chain, and it's, again, you get it kicked off once the amino acids it's like a magnet that bond to each other. So step 1, codon recognition. Step 2, more transfer RNAs come to bring more amino acids. Step 3, one gets kicked off when we just kind of slide down the chain here. And we're just, with each transfer RNA, brings a new amino acid that bonds to the chain of amino acids and then kicks someone off, just bumps off the chain. And in the end, we get a useful protein in the end. And we'll show you sort of a nice video that kind of goes through that process. But here again is the base, and we're going to work hard on reading that genetic code because we want to, given the 3 base messenger RNA code, we can figure out what amino acids should match with that. So these are your notes on protein synthesis. We will do lots of practice in class.